Okay, hello everyone. So this is going to be the first, I'm just going to do the first part of the lecture, which basically is a little bit of introduction and then pertains to the Hardy-Weinberg, because then you guys have a few problems, probably 3 problems that you'll work on together to do in class on Wednesday, and then either hand them in to Dr. S on Wednesday or hand them in to me by Thursday. They just have to be to me by Thursday. You can work in groups. If you work in groups, you all can hand in one assignment together. And then if you have questions on that, I will be in my office on Wednesday from like 11 to about 12:45. So any questions you might have when you guys are doing them, you can ask me then, okay? And then the second part of the lecture, I'll record as well, and I'll put that up by the end of the week. You'll have a quiz on Moodle to do with that. There will be no calculation questions or problems about pertaining to Hardy-Weinberg on the quiz, okay? So you'll have intro on Hardy-Weinberg now, and then the problems that you're going to do related to it, and then you will have a quiz in the second half of the chapter, which is just, theory, talking about natural selection, different types of natural selection, and other ways of sort of introducing genetic variation into populations. Okay, so this chapter is population and evolutionary genetics. These are the topics that are covered in the chapter, and like I said, I broke it up into 2 separate ones. Really, this first lecture is going to stop here after this section 22.3. Okay, and then the second recorded lecture will be the last part of the chapter. Okay, these other sections here. Okay, so just a little bit of introduction here. So we've talked about evolution before. We talk about it in, Bio 113, and actually even at the end of Bio 111. We probably covered some of this stuff in Bio 111 towards the end. Actually, we covered most of this, maybe in not as much detail, but you guys all should have heard of this before. So obviously, we know that evolution is a consequence of changes in genetic material, right? And so basically over time, we have genetic changes that occur that lead to the formation of different species, right? We talked about Darwin's theory of evolution and natural selection, and that natural selection drives evolution. And I actually think this is sort of duplicated a little bit here. Yeah. Well, sorry, I didn't realize the book had this little intro in there, and I already added it in on the next slide. So sorry, there's some duplications here. So let me just switch to my slides that I had added in where we first started talking about the first section. So don't let me confuse you. But what is in those previous slides, marked introduction, that was already inputted in by the book, and it's essentially the same thing here. So just to sort of reiterate, right? When we talked about Darwin and also Wallace's, sort of idea of natural selection, they knew that natural selection existed and that it drove evolution, but they didn't know where the origin of variation came from, right, within a population, right, or how those traits were then passed on. They just knew that there were changes that occurred that were fueled by the environment that eventually lead to the formation of new species. Then, we've talked about Mendelian genetics, and so basically kind of the conversion of Mendelian genetics and also the idea of natural selection sort of converged to basically form this sort of branch of genetics called population genetics, okay? And so basically in population genetics, geneticists are going to study the gene pool populations to see what types of variations are there, right? And that may then lead to speciation. So not all the time, right? So you may have changes in a population or a lot of variation in a population, and it may not end up producing new species. However, it also may end up producing new species. So basically, you'll have a change in a population that's going to occur first on a smaller scale that will then eventually end up leading to the formation of a new species, right? And so you kind of talk about the difference between microevolution, right, which is here, genetic change within a population, right? Same species, a population is composed of all of the same species. That will then, those changes may or may not lead to macroevolution, right? Where you're then going to talk about the development of new species. So what Darwin talked about really was macroevolution, right? He was observing all of these new species that occurred and talking about theories of evolution and how the environment affected it. However, he had no way of analyzing the alleles in the population or knowing how those traits were passed on, okay? So what we're going to be focusing on in this chapter really is microevolution and population genetics, okay? And so as I said, if you talk about a population, it is a group of individuals belonging to the same species. They all live in the same geographic area. It says they actually or can potentially interbreed. So bottom line is they can interbreed with each other because they are the same species. If we talk about the gene pool, okay, those are all of the alleles that are present in a given population. Okay, and so what we're going to talk about here is really focusing on, two alleles, right? So if we talk about any one given gene, right, we'll talk about a dominant and a recessive allele, and so that's the way we're going to talk about the population in terms of the alleles that exist, okay? It says that most populations contain a very high degree of heterozygosity, right? So bottom line is, that's going to provide the genetic variation, right? So if all the individuals in a population were all homozygotes or all homozygous recessive or homozygous dominant, you're going to lose some of that variation, right? Because maybe the dominant phenotype is favored, right? So you need to you have that dominant phenotype make you fit for that environment, but there also are going to be many individuals who are carriers and can carry that heterozygous allele, okay? Again, that lends to that variation. So if we were all homozygous, so it's really more thinking about us all being homozygous dominant, having the normal alleles, right? And then having no variation in there, okay? There is, in reality, a lot of variation that occurs in a given population, and for very good reason. Okay. Now, one of the ways you can talk about genetic variation, or one of the ways to sort of detect genetic variation, is by artificial selection, okay? And so you can basically visualize this with one good example, dogs, right? Different breeds of dogs. So basically, it says, so let's just kind of talk about this, right? So it says if genetic variation does exist, then a phenotype will change over generations, right? So you'll see the phenotype change because of altered alleles, or recessive alleles, and basically, if you're talking about the whole, organism in general, there's going to be multiple different recessive alleles or other alleles with amongst all the genes, okay? That's going to lead to variation, right? So if you just look, for example, at the size difference between a Chihuahua and a Great Dane, you can see how there's a huge amount of genetic variation within that species, right? And if you selectively breed, you can pull out these sort of different specific traits and create these very different looking, phenotypes, okay? That's essentially what these different breeds of dogs are, okay? They're different phenotypes, okay? Again, all from technically the same species, right? So that's a way of sort of this artificial selection, and the way we've artificially selected different breeds of dogs is a way of sort of visualizing the amount of variability that exists, okay? So that's the whole point of this. Obviously, now we have many different types of technologies, right? DNA technologies that allow us to analyze alleles and genotypes within a population, okay? So we can directly look at nucleotide sequence, right? So here's one example. So if you talk about individuals within a population and you look at the gene that's going to encode for an enzyme alcohol dehydrogenase, okay? Basically, this was in a population of Drosophila, so they studied, the study that was done, isolated and cloned and basically sequenced, right, copies of this ADH gene, right? So they were basically looking for different variations within that gene. And so again, like I said, we have technology that we can actually look at the nucleotide sequence, right? We don't need to selectively breed or artificially, use artificial selection to try to figure it out. We can actually visualize the DNA because of the technology we have, right? So the next figure here is figure 22.2. I'll explain it a little bit more in terms of what it's showing, right? So like I said, they analyzed this ADH gene in Drosophila, okay? And basically what they found from their study, right, with smaller scales, that they actually found that there's two different alleles, okay? That you can have this sort of ADHS version and this ADHS version, okay? And so basically it's a difference, one of them contains this switch between an A and a C, right? So it's called an AC polymorphism, and you'll see this denoted in the next figure with an A and an asterisk, okay? And so that's what that's showing you. And so basically, because of this single nucleotide change here at this one specific position, you would get different proteins, right? Because that change basically would encode for a different amino acid, right? So you're talking about having a threonine versus a lysine at this codon 192, okay? So again, based on the nucleotide change, you can change the amino acid that's coding for, therefore change the final version of that enzyme or that protein, okay? Which then would lead to some sort of phenotypic or functional change. All right, so again, they looked at different populations. They had about 5 natural populations of Drosophila that they cloned. And then from that, they found about 43 different nucleotide variations, okay, in this ADH sequence that they studied. Out of those variations that they found in coding regions, only actually 1 led to an amino acid replacement. So again, you can have changes, right, nucleotide changes, that may then still code for the same amino acid, right? So if they still code for the same amino acid, the protein's going to be exactly the same, right? However, they did find that 1 will actually lead to a change in the amino acid, like I mentioned above, right? And so if you look here, so this is showing you this ADH sequence, right? You have exon 3, intron 3, and exon 4, and you can see here, this is the position where you have this A to C change here, all right? Sorry, this A to C change here, right? The A with the asterisk here. And so if you look down here, you can see, or where are we? Here, this change. Hello, if I could line it up, right? You can see the dot means it's the same, still an A here, but in these, you can see that instead of the A being there, there's a C, okay? And so wherever you see dots, it means that, and these are the different, strains they looked at, different populations. Wherever you see dots, it's the same nucleotide. Wherever you see another nucleotide, right, it means there's a change. However, a lot of these changes didn't lead to the coding of a different amino acid. Therefore, you still have the same protein or the same enzyme coded for, okay? It's only this one position here where these changes led to a different amino acid being coded for, okay? Now we have even, more technology than that. We have different types of sequencing technology now, which has basically automated the process and made it way easier to be able to sequence entire genomes, right? So there's basically a sort of project going on that I guess was started in 2008, and there's other projects like this too where it says it's the 1,000 Genomes Project. Bottom line is we're trying to sequence as many genomes as we can, okay? And so this is saying, and so we've done that with many different organisms. We know the genome, right? This is now talking about an effort to basically catalog 95% of common genetic variations carried by about 7 billion people, okay? And so basically what they're trying to do is associate specific genetic variants with disorders, right? And so some of the pilot data, I guess, that's come out of this has identified about 15 million single nucleotide polymorphisms, amongst other data as well. But I just wanted to make sure I mentioned single nucleotide polymorphisms because you hear this a lot, and you also see this abbreviation SNPs. And so all that means is it's a change, a single nucleotide change, just like we talked about before in the Drosophila, right? Where you had to change from an A to a C that then led to the coding of a different amino acid, which then affected that final protein or that final enzyme. It could lend it dysfunctional. It might make it have increased function. You don't really know, right? But bottom line is some of these polymorphisms, and again, other genetic variations, can then be correlated or related to disease, right? And so then we can basically, one, predict disease, and then also maybe use the genetic information to be able to treat disease more specifically, okay? And so a lot of cancer treatments and treatments for other diseases as well, kind of trying to move more towards this individualized medicine. And you obviously need genetics to be able to do that, right? So if you have this genetic change, you should be treated in this way, versus somebody else with the same disease might have a different genetic change that's causing the disease and therefore should be treated in a different way. Okay, so you can see how it's so important to understand the genetic basis of disease. And most of the diseases we're talking about are not really pinpointed to one specific loci, but it still gives us some information of sort of relatedness, right? So it's very complicated. Some diseases are, right? We talk about purely genetic diseases, and you guys probably talked about that early in the semester. But this is even helpful when we're talking about something complex like cancer, right? Where there are many, many genes involved. Okay. So what we're going to talk about next is HardyWeinberg. Again, we mentioned this. We talked about this in Bio 111. We didn't really do much with it, and we're not going to go crazy with it here, but I want to give you guys a few examples, and then, like I said, I will have problems for you that you will get on Wednesday in class, which Dr. S will give them to you. You will work on them together in class, complete them, and hand them in. Okay, so what is HardyWeinberg, and why do, population geneticists basically use it? I mean, it's a way to determine how much, oh shoot, that's a typo, obviously. Let me fix it right now. Can I do that? I think so. Not how much genetic population, but how much genetic variation, right, is present in a population. So we fix that, right? So how much genetic variation is present in a population? Are these genotypes randomly distributed? Are there patterns, right? If you look at a population over time, you can, analyze it at, say, year 1 and then at year 10 to see how things are changing. And maybe it can tell you something about the environment, right? So there are many, many applications of, population genetics. Maybe you're interested in this if you're developing a new species, but maybe not, right? Maybe you're interested in what the impact of the environment is on this population, right? And maybe there are things we can do to prevent some of these changes, right? So basically, the Hardy-Weinberg law, or the Hardy-Weinberg theory or equation, whatever the heck you want to call it, it basically describes what happens to allele frequency and genotype frequency in ideal populations, okay? So there's a few sort of things that are criteria that a population needs to meet in order to, be termed ideal here, okay? And so they're outlined in the next slide, or next couple slides. I'll make sure I go back to that and point it out in a second. Right, so some of these things are here, but again, I have them sort of numbered out in a more clear way, okay? What makes an ideal population, right? So it says infinitely large, so you have to have completely random mating, and there is no other evolutionary force acting on the population. No mutations, no migration, no natural selection, right? And so you can see that this is not true of most populations, but we basically use this as criteria then to see how much things are changing, right, in a given population, right? So if it deviates from this, then that means that there's evolution occurring, okay, from this sort of Hardy-Weinberg equilibrium. Okay. Right, so again, if you just look at this model and you talk about just a single autosomal allele, right? So autosome, non-sex chromosome, 2 alleles, your dominant allele and your recessive allele. Okay, and this is, again, just a simple example. Frequency of your dominant allele is 0.7. Okay, this is in a given population. And if this is an ideal population where you don't have any mutation, you have completely random mating and all these things occurring, this is how these numbers are going to work out, right? So your frequency of the dominant allele is 0.7. Therefore, the frequency of the recessive allele has to be 0.3. Okay, so these always have to equal 1. Now, if you talk about what's the frequency or the probability of being homozygous dominant, right? It's 0.7 times 0.7, okay? Because we know for each allele, the frequency, the probability is 0.7. So we multiply it, and that's 0.49, okay? So that means that a homozygous dominant genotype will occur 49% of the time, okay? If you talk about the frequency, the probability of being a heterozygote, okay, it's 0.7 times 0.3, okay, times 2, or 40.42 or 42%, okay? If you talk about little a, 0.3 times 0.3 or 9%, okay? So let's look at that figure and kind of look at this sort of out in the Punnett square, because we've all seen Punnett squares before, okay? So hopefully this should look familiar, okay? So this is showing you here, so if you're talking about male, right, male gametes, versus female gametes. And so the frequency of, again, of being, right, of having the dominant allele versus the recessive allele, right? So in any one gamete, okay, again, we're only just talking about two, there's only two alleles here. And so here's your 0.7, your 0.3, your 0.7, and your 0.3, right? We said this before. So male, female gametes, okay? And so now if we look at what's the frequency of being a homozygous dominant, 0.7 times 0.7, right? Or 0.49 or 49%. If we talk about the frequency of being a heterozygote here, right? Because now you're getting, because here's the, right? Here's the thing. This is why we multiplied by, or, we multiplied this by 2, right? Because you're getting the recessive allele from the male, right? Here, and then you're getting the dominant allele from the female, right? And so again, that probability would be, or that, you know, the frequency of this would be 0.21. And if you look vice versa, whether if you're getting the recessive allele from the female, right, and the dominant allele from the male, 0.21, okay? And so therefore the frequency of being a heterozygote, because again, it doesn't matter what order it's in, right, or where it comes from, whether it comes from the male or the female, it would be 0.21 times 2, right, or 0.21 plus 0.21, okay? And so that's where we got, if we go back here, that's why we got this, right? We multiplied by 2, right? 0.21 plus 0.21 is 0.42, or 0.21 times 2, okay? That's where that comes from. If you now go back here, if you look at the frequency of being a homozygous recessive, right? Again, you're getting the one recessive allele from the male, one recessive allele from the female, 0.3 times 0.3, 0.09, or 9%, okay? And so again, remember, this is all, you know, based on, well, I'm going to show you the equation in a second, right? So it's all based on essentially this equation here, okay? So like I said, I told you I was going to sort of outline more of these assumptions, right? Because we're saying this isn't an ideal population, okay? And so we call this, if you have this all equaling 1, right, it's basically at Hardy-Weinberg equilibrium, okay? So it says allele frequencies in our population do not change from one generation to the next, okay? Basically, you can calculate this because this population is in equilibrium. Again, probably not the most case realistically, but we use this model then to see how much evolution, or to what degree microevolution is happening in a population, okay? So again, if you want to now add letters to this, to what we just did before with numbers, okay? Lowercase p will equal the frequency of allele of the dominant allele, okay? So that's this guy here, right? So again, if we go back here, the frequency of the dominant allele, 0.7, right, is equal to p, okay? Q is the frequency of the recessive allele, okay? So again, if you go back, frequency of the recessive allele is Q, right, which is 0.3, okay? Oopsie. All right, and so now, if we now, instead of using numbers like we did in that last example, now we put the letters corresponding to it here. This is what we come up with. So it's probably a good idea as you're reviewing this to kind of compare the same Punnett square with the numbers versus the letters, okay? Again, these then fit into that equation, that Hardy-Weinberg equation that I just showed you, okay? So again, if you're talking about the frequency of the dominant allele, that's going to equal lowercase p. If you're talking about the frequency of the lowercase allele, it equals Q, okay? If now you look at genotypes, right? So if you're talking about the frequency of the homozygous dominant, okay? Genotype, you're talking about P squared, right? This times this, the frequency of this times this, right? That's where this comes from. If you now talk about the frequency of the heterozygote, again, it's, right, P times Q, right? Here, same thing if you're looking, because now if instead of the, right, instead of the, whatever, the dominant allele coming from, so here, the dominant allele came from the female, here, the dominant allele is coming from the male, okay, again, Q times P. Remember that but now it doesn't matter which order these guys are in. So 2PQ will give you the frequency of being a heterozygote, okay? And then now, if you're talking about being a homozygous recessive, okay, it's Q squared, right? Or Q times Q, right? Or in our example, 0.3 times 0.3. Okay, so that's how that all matches up. So make sure you understand that before you go and try to do the problem. Okay, all right, so here are the assumptions for Hardy-Weinberg. Okay, there's an equal rate of survival and reproduction, and there is no selection in this population. Okay, there are no new alleles that are arising within the population because there's no mutations, right? So none of the alleles are changing. No migration into or out of the population. The population is huge, right? And also random mating is occurring. So all of these things, if these are not the case, will affect this Hardy-Weinberg, okay? And then that population would not be at equilibrium, okay? Again, we're using this as a model. Okay, there are also sort of 3 additional things that also are part of Hardy-Weinberg, okay? It says dominant traits do not necessarily increase from one generation to the next, right? So you're not going to see, again, you're not seeing a change in alleles, right, frequency. So you will not see more dominant traits appear because that's sort of the favored phenotype. Because in this case, it's not. Genetic variability can be maintained, right? So again, the allele frequencies will remain unchanged, okay? I kind of said this already. If you know the frequency of one genotype, you can calculate the frequency of other genotypes, okay? And that's what we did, and that's what you guys are going to do with these practice problems, okay? So the first practice problem that you're going to do in class is a mystery, okay, is actually going to be number 1 on page 45 of the book. Oh, it's not 45. Look, see, so I screwed up again. It isn't page 45. It is page, what page is it? Oh, gee. One second, let me get the page here. Yeah, it's page 445. Okay, so let's fix that too. Sorry about that, but we fixed it now. Okay, it is page 445 and it's 22-1. That'll be the first practice problem that you're going to do. Okay, and so again, it's asking you questions based on what I just showed you with that last example. Okay. All right, now, obviously, we want to use this sort of tool to basically figure out whether evolution is occurring or what the frequency of alleles is in a given population. We want to apply this to human populations, right? And maybe apply it to disease in some way. And so one other example, so like I said, that last example, that last problem is really just a straight kind of look at, here's a given population, or here's, a given sort of frequency of a certain allele or of a certain genotype, and then you have to basically figure out all those other terms, right? So if I tell you what P, or you figure out what P is, you can then calculate what Q is, okay? Based on that equation. That's what that first problem is going to be. This next example, again, is more applicable to the human population and to disease, okay? And so for this one, we're talking about susceptibility to HIV-1 infection. And this has been sort of susceptibility of even developing the HIV infection to begin with has been based on a gene called CCR5, okay? And this gene is actually codes for a chemokine receptor, okay? So it's called CC chemokine receptor 5. And chemokines are basically signaling molecules that are associated with the immune system, okay? They also, the receptor, this chemokine receptor, is actually the receptor on the cell that HIV-1 uses to gain entry, okay? And so if you have a mutation in that receptor, the HIV actually will not be able to get in to the cell, which obviously in this case is a good thing, right? So having the mutation of this chemokine receptor is actually a good thing in this case. Okay, so let's look at this a little bit more closely. I'm sorry, I don't know why that's coming up. Okay, right, so if you look at this figure 22.5 in your book, it's actually showing you the CCR5 gene, right? and showing you the exons here. And so if you look at this one particular exon, exon 4, there's actually, what we found is that there's this mutant allele, right, that basically has a 32 base pair deletion here within this exon. And that mutation, right, or that deletion will block entry of HIV-1 into the cell. Okay, and so again, individuals that have that mutation actually are protected, right, in some way, right? So if you look here, homozygous individuals resistant to HIV-1, so meaning homozygous for that deletion, right, that delta 32. I'll show you a table in a second. If you're a heterozygous, you're susceptible to infection but progress more slowly. Okay. And obviously, if you're talking about just being a normal dominant allele, right, you don't have that deletion in that CCR5 gene at all, right? You're just normal. You don't have the mutant allele. You call that basically the normal allele. Okay. And so basically, you don't have any protection. Okay. Sorry about that. I have a few little interruptions here. So let's just finish this now. All right, so we're talking about resistance to HIV, right, and the involvement of this chemokine receptor. So let's look at this figure. So it says the genotypes were determined by DNA analysis using PCR, and then also restriction enzyme digest. So basically, we're able to determine if an individual, either was a homozygous for the normal allele, or heterozygous, right, containing that deletion in one of the alleles, or if both alleles contain that the deletion in that chemokine receptor gene. Okay, and so if you look, we want to look at this first, I guess, huh? Because I guess I didn't really explain this fully. So this is what I just mentioned, right? So if you're talking about your genotype, 1 meaning the normal, okay, CCR5 gene. So that means if 1/1 meaning you're homozygous dominant, essentially, think of that, or you're homozygous for the normal allele, okay? And so obviously you would be highly susceptible to developing HIV because the virus will be able to gain entry into the cell easily. However, if you're a carrier, right, for the 1 mutant allele, right, so that, again, it's this delta 32 means this 32 base pair deletion, right? You may be susceptible, but it actually may progress to AIDS more slowly because the HIV might have a little bit of problems gaining entry into the cell. If you have both of those alleles or that mutant allele with that deletion, you actually are resistant to most of these HIV strains. Okay, and so obviously it's probably advantageous to know whether or not you have that mutant allele or if you have both mutant alleles, what have you, right? And so again, that kind of answers this question, right? Which human populations carry this allele and how common is it, right? So is that allele really common in the population? So obviously you can take samples from a population and see how many of the mutant alleles actually exist, right? And again, once you figure that out, you can calculate what the frequency of of this homozygous mutant genotype is within a given population. And if it's different, if you look at different human populations or different parts of the world or what have you, okay? So bottom line is, like I said, they use PCR to basically amplify DNA, to amplify basically a specific region of that CCR gene using specific primers. And then they also use restriction enzymes to cut at specific points to basically get these fragments, right? So they basically know if they see this 403 base pair fragment and a 332 base pair fragment, that this individual has both normal alleles. If they see this 371 base pair fragment and also the 332, then this person has both mutant alleles, okay? However, if they see three fragments here, a 403 base pair, a 371 base pair, and a 332 base pair, this individual is a heterozygote, okay? And so they're able to basically genotype these individuals, right, for this CCR5 gene, okay? And again, this then correlates to susceptibility of developing HIV, or basically of that HIV virus being able to enter into their cells. Now, if you then use HardyWeinberg equilibrium, right, to basically further study this. Like I said, now once you know one of those frequencies, right, you can then basically calculate the other, right? So if you know the frequency of the mutant allele, you can then calculate the frequency of the normal allele. You can then calculate what the frequency of being a heterozygote is, okay? Because you have this Hardy-Weinberg relationship, okay? And so again, if you look at this table here, okay, it's just showing you two different methods of determining these allele frequencies, right? So basically you can count the number of alleles, but you can also figure this out from genotype frequencies here, okay? And so again, this is all based on that Hardy-Weinberg equation here, or relationship, and you can basically solve for whatever you want. And so your next homework problem, or one of the homework problems you're going to do, will involve this table, okay, or not homework problem, I guess in-class problems, and some of these, sort of frequencies, and I'll probably change up the frequencies that are described here, and then just ask you to, calculate maybe the frequency of this CCR5-1 in a sample, right, which we're calling the sort of normal allele. And then this is showing the frequency of this delta 32. Remember, that's your mutant allele, right? So given this, what they have here, right, the frequency of the normal is 178 out of 200, or 0.89, right? Because it's showing you here the number of, right, of one alleles, okay, that is basically 178 in total, right? The number of total number of alleles is 200, okay? And so if you want to know the frequency of the CCR5-1 allele in the sample, it's 178 divided by 200, or 0.89. If we talk about the frequency of that delta 32, right, the deletion, the mutant allele, you see that there are 22, right, alleles, delta 32 alleles are mutant alleles out of the 200 total, okay? So 22 out of 200 is 0.11. So basically, I'm going to ask you to basically do these calculations, but with different numbers, okay? So we're going to make sure that we understand how to do that. And then there'll be another question that sort of is related to this, but asks you slightly different. But again, all relating back to this equation, okay? And so again, make sure you review this before coming to class on Wednesday. That way, it makes it a little bit easier to do the problem set. Okay, so again, and so let's kind of continue here, right? So if you look down here, right, we already knew this, right? And this is basically, it's basically showing you, this is basically showing you if you're counting alleles versus using genotype frequencies, that you're still going to come up with the same numbers, because again, everything is based on this, right? So remember what P stands for, right? And what Q stands for. If you go back to our initial example with our little simple Punnett square, it's the same thing, okay? So remember, it all goes back to that. We're just going to be all of our problems are just going to be involving two alleles, okay? Though I will just mention that you can do this with more than two alleles, right? So you can modify the Hardy-Weinberg equation to include additional variables, right, for 3 alleles. So if you're talking about something like blood typing, okay? I'm not going to ask you to do a problem like this, but I want you to know that you can modify it this way. So basically, in this case, you don't only have P plus Q, you also have R, right? But again, they all have to add up to 1, okay? So P plus Q plus R equals 1, okay? And so then if you look at the genotype frequency, you got to add in a whole nother, term here, okay? So instead of just having, you got to add in the R to this, okay? So it makes it a little more complex, but in actuality, it's not really that big a deal. It's just more numbers. So like I said, you would use this in reference to like blood typing, right? So you have multiple alleles there if you're talking about A, B, and O blood types, right? This is what the genetics will look like, right? So if your genotype, if you have 2 big A's, right, your phenotype is A. You can also be A if you only carry one of those A alleles, right? 2 B's or 1 B will give you B genotype, but then you can also have A B. This doesn't make you, right? It's not that B is recessive to A or that you have big A, little a, right? These are all basically codominant, right? We call this codominance. And so you can see how this is different than just having 2 alleles, 1 dominant, 1 recessive. Okay, but you still can use Hardy-Weinberg to assess this population. Okay, it's just that you have to add in some additional terms here, right? So I said basically, if you want to kind of try this out, if you don't believe me, you can insert the numbers from the table, right? And then insert them into this equation and see if you get close to 1. Okay, so if you want to try it out, you can try it out. Like I said, I'm not going to give you a problem where you have multiple alleles. We're going to stick with just a dominant and a recessive allele for our so again, just another application here, right? So it says HardyWeinberg allows estimation of frequency of heterozygotes in a population. So again, this is kind of, you're talking about a genetic disease like cystic fibrosis, right? You're interested in knowing how many carriers exist in a certain population. And again, you might see that, you know, in certain populations there are more carriers than in others, and then that might, you know, sort of prompt you to ask, well, why? Right? And so there are many instances where you have, you know, protection as a heterozygote. We actually kind of, right? And so if you talk about like sickle cell anemia, right? If you're a carrier for that, protects you against malaria. Okay, so there's many reasons why you want to know, you know, how many heterozygotes are there, or how many recessive alleles are in the population, and basically, you know, you might compare it to another population or another, you know, that same population at another time, and then again ask questions about why it's changing. Okay, so like I said, example, cystic fibrosis, right? So this is an autosomal recessive disorder. So unlike, you know, when I was talking about complex disorders like cancers where there's multiple genes pinpointed to it, we know cystic fibrosis is, you know, purely genetic and pinpointed to one specific gene, right, that affects a chloride channel. So this is basically telling you so that you have an incidence of about 1 in 2,500, right? So about 0.0004 people in Northern European ancestry that have cystic fibrosis, okay? And so if you look and use HardyWeinberg to basically figure this out, right? You can see that you get to this 4%, right? In terms of the frequency of the recessive, okay? So anyway, it's a way, again, it's a tool to use to determine, you know, how many recessive alleles there might be in a given population. And again, that might then prompt you to ask more questions from there. Okay. All right. So that's the last slide here. Like I said, I will upload another new recorded lecture for the rest of the chapter. That will just have a Moodle quiz. There are no calculations or problems. Okay. On Wednesday in class, you will basically have like 3 problems. There's not going to be many. It's not busy work, but you know, a couple of them I already kind of told you what questions I'm going to ask you, because they're just going to basically they're going to come from the book. They're just basically example problems. Okay, so you guys can work on them together or individually. Make sure you hand them in either to Dr. S or to myself. If you have questions, you can ask. Okay, bye. Thanks.